Meiosis. Meiosis is a two-division process that results in 4 cells from the original mother cell. Meiosis is divided into 2 phases, meiosis 1 and meiosis 2. Only certain kinds of cells participate in meiosis, and those are animal cells, the gametocytes, and the life cycle of that would be the adult undergoes meiosis or reduction in the genetic material so that you get 4 gametes with half the chromosome number, or N. These would be haploid cells. 2 haploid cells fertilize and become a zygote with the 2N, or the full set of chromosomes needed for the zygote to develop normally, grow into a 2N adult. In higher plants, meiosis can occur in alternating generations. Here you have the zygote that grows into a sporophyte. These are both 2N, full set of chromosomes. The sporophyte, the mother spore, undergoes meiosis and has the haploid cells, 4 haploid cells from the 1 2N cell. These spores develop into gametophytes, and those gametophytes form gametes, which fertilize and become the zygote, which is the 2N full set of chromosomes. Back to the sporophyte again. So this is the 2N portion, the diploid portion of the plant cycle, and this is the haploid portion of the higher plant cycle. And that's why it's known as alternating generations. Before meiosis begins, just like in mitosis, the DNA has to replicate. Once the DNA has replicated, meiosis 1 can begin. Meiosis 1 is divided into 4 phases, similarly named as with mitosis: prophase 1, metaphase 1, anaphase 1, and telophase 1. In the first prophase, the chromosomes condense, the nuclear envelope disappears, and crossing over can occur. You can refer to your worksheet on crossing over for that. When you compare meiosis and mitosis, you can see that the tetrads are together, whereas they are separated in mitosis. In meiosis, the tetrads, the homologous chromosomes, all come together and form. In metaphase, again, pairs of homologous chromosomes are going to move to the equator, and they're attached to the spindle. This is meiosis 1. You can tell because there are tetrads. In mitosis, you can see that the homologous chromosomes are separated out, and you have 4 along the equator rather than just the 2. In anaphase 1, the homologous chromosomes separate. They're pulled to opposite sides by the spindle, just like in mitosis, but the chromatids do not separate. In mitosis, the sister chromatids are separated. In anaphase, the chromatids are not separated. Each chromosome still has 2 chromatids. one analogy could be that the mother's chromosomes all went onto one side and the father's chromosomes were all pulled to the opposite pole. And then in telophase, cytokinesis happens and you end up with two cells, each having one chromosome from each pair of homologous chromosomes. So each cell has a complete set. It's a 2N. Before meiosis 2, the DNA is not duplicated. So a new spindle forms around the chromosomes that are from the end of meiosis 1. Again, in metaphase, they line up at the equator, and here they're still at the 2N phase. Anaphase 2, the centromeres divide, and the chromatids are separating. And now each individual chromatid is called a chromosome, and they're moving to the poles. At the end of telophase 2, you have a nuclear envelope forming around each set of chromosomes, and cytokinesis happens, and you have 4 daughter cells, each with a haploid condition, half the number of chromosomes needed for an organism. What's the significance of meiosis? Well, diploid, the 2N number of chromosomes, is reduced in such a way that you have 4 daughter cells, each with a haploid set of chromosomes. And that's important in sexual reproduction. And when you have the 2 gametes come together, the 2 haploid gametes come together, and you have a 2N zygote. For a normal organism to develop. The second significance of meiosis is the randomness of the process of chromosome separation. So we have 23 pairs of chromosomes. So you have 2 to the 23rd possibilities for chromosomes. That's 8,388,608 combinations for chromosomes. And then you can have recombination. You can have crossing over. You may see your worksheet on that. And independent assortment of homologous pairs. So that's where each pair can move independently of the other pairs. To further clarify that, that's called Mendel's rule of segregation, and we'll learn more about Mendel coming up next. Each gamete receives 1 chromosome from a homologous pair. And in anaphase 1, that's when this separation happens. The direction of separation is independent of the tetrads. So you could have 1 chromosome from your mother and 1 chromosome from your father, or a different chromosome number 2 maybe from your mother and chromosome 3 from your father. So there's an independent assortment of the chromosome pairs that recombines into a unique individual for each gamete.